Okay, so here's the repeat of yesterday's lecture. I was not happy with yesterday's lecture at all. I think what happened was I'm doing things in a different order than your textbook, and I kind of got lost between what I've done before and what's in your textbook. So came back, reorganized the whole thing. We're ready to rock and roll now. So we're going to start on page 3 of the chapter 3 notes, which is something I did on Monday, reviewed on Tuesday or Wednesday, but that's just a good place to start for this. So I'm going to share my screen, which I know how to do with this Zoom, as it turns out. All right. So this just lays out what the ARPs, actin-related proteins are that are involved in polymerizing straight microfilaments. Okay, how do you make it? And keep in mind here we've got both polymerizing enzymes and depolymerizing enzymes, because whenever you change microfilaments, it's a dynamic thing. You're breaking down here and you're putting it together over there and you're changing the cell shape. So we've got both things going on here. So the first thing is kind of when cells are quiet, you've got Cap-Z on the plus ends of the microtubules or microfilaments, and that's going to block depolymerization, so you're stabilizing it. And that Cap-Z will also bind to other structures and keep the microfilament attached to the rest of the cytoskeleton. We'll see this in skeletal muscle sarcomere structure on Monday, when we see that Cap-Z is bound to the Z line, and there are intermediary filaments there that it sticks to. So Cap-Z is protecting that plus end, but also binding the microfilament to something else. Thymosin beta 4. So the idea here is that at the concentration of actin and ATP in the cell, they would spontaneously polymerize, and that would be chaotic. So thymosin beta 4 basically sequesters those monomers, so they're not interacting with each other until the cell needs them to polymerize and change the shape of the cytoskeleton. Third thing here is profilin, which is going to take these monomers of actin, G-actin, from thymosin beta 4, take them over to the polymerizing plus end of a microfilament, and give them to number 4, formin, which then adds them to the chain and makes them longer. Now, formin can do this polymerization without profilin, but it's stimulated quite a bit by having profilin there. And we'll talk about that more in a second. So that's all the polymerization part, 2, 3, and 4. And then for the depolymerization, we've got 5 and 6. So 5 ADF cofilin is going to depolymerize at the minus end. You'll see how important that is when we talk about lamellipodium. At the minus end, we're depolymerizing. At the plus end, we're polymerizing. And then gelsolin, which is going to cut in the center of a microfilament, and that provides more minus end, so you can depolymerize faster. So those proteins are all going to be activated at the same time, and that's going to stimulate polymerization of the microfilaments. Here's this animation. I don't want to play it again, but you should watch it if you haven't seen it. It's really cool. The big green thing here is the formin. And formin in this video, talk about this a little bit later, but formin is going to be activated by a G protein called Rho, and it then binds itself to the membrane, gets ready to go, and then starts to grab G-actins and put them together and polymerize them. And it goes kind of slow at first because it has to nucleate. Once it nucleates, it goes a little faster. But what really speeds it up, you'll see, is when the orange guys here, the profilin, start to become involved, because profilin will bind to the monomer. It'll also bind to the formin, and that helps to bring it in and organize it in the right order and the right shape, so that it's going to fit up in there, and it polymerizes really quickly that way. So that's for polymerizing long straight chains of microfilaments. And that's most microfilaments are that. When you get into the lamellipodium, which we'll talk about today, you've got branch microtubules as well. So how do you do that? Keep in mind, whenever you're polymerizing, you have to start from some nucleation point. So if you got a long chain, how do you branch it? You don't have a nucleation point. Well, this process here is going to help to nucleate it. So we've got three different proteins, well, really four proteins here that are involved. CDC42 is a G protein, a small monomeric G protein, one of the Rho GTPases that we'll talk about today. And that G protein, when it's activated, will bind to WASP and activate WASP. WASP is then a protein that will grab onto ARP23, take it over to the membrane and attach it to the side of a microfilament, and that'll form a nucleation point on the side of a microfilament, and it takes off at like a 70-degree angle from the original microfilament. And you can see that in this animation, which is really cool. Again, same people made this animation as the last one, and it's absolutely gorgeous in my opinion. What's really neat about this animation, you can see this green thing here is the WASP. And you can see this is it's just a classic way that molecules activate and deactivate. So WASP is kind of like folded up on itself because it's binding to itself here. And then CDC42 comes in and binds to that same site, and that releases this site. And now this site can grab onto ARP23, and it brings it over to the microfilament and attaches it to the side of the microfilament. So basically, just a really, really gorgeous animation. Here's more of a figure. This is again from that World of the Cell text I use from time to time. So, oops, damn it. WASP is bringing A2 over and attaching it. Hey, Brittany. Oh, is Brittany here yet? Hey, Brittany, I just got started. So I'm only about 5 minutes into my spiel. So you can catch up right now. All right. So again, that's something I talked about Monday. I talked about it again on Tuesday. So I'm just reiterating here. Cell crawling. Okay. So what we're leading to here, what we're eventually going to get to is cell crawling, which is how non-muscle cells move. And what that has to do with a lot is chemoattraction. So what I'm trying to say is what we just talked about is the polymerization of the microfilaments. Now we're going to get into why they're changing and how they're changing. Excuse me. So usually what we're talking about with cell crawling is some kind of chemoattraction. So there's some chemical out there, this little blue guy here is a hormone, which is then going to be binding to and activating receptors, which will activate single transduction that will then stimulate this polymerization that we've been talking about. So two things I want to talk about here, they both involve PI and PI turnover. So this is PI over here, which is, I think this is slide 4 of chapter 2 PowerPoint. This was on there. So this is just a phospholipid, phosphatidylinositol. Let's see. You've got your glycerol backbone here, 2 fatty acids, one there. And then you've got, here you can hear you. Okay. And then you've got a inositol here, which is basically cyclohexane with an OH on each carbon. And then it's phosphoric acid bonded over here to this. So that's your PI. Now, this is a really cool phospholipid in that it's used for signal transduction a lot. This is just one example of how it's used. Over here, these phosphates, if you number these carbons, it's 1, 2, 3, 4, 5, 6. So there's an enzyme here, PIK, which is phosphatidylinositol kinase. It's going to come along and put a phosphate in the 4 position and in the 5 position. And that's how we get PIP2, which, technically its name is phosphatidylinositol 4,5-bisphosphate. So we're putting phosphates on the 4 and 5 position here. And what that's going to do then is you've got this phospholipid with these big negative charges sticking out there. I think, I'm not 100%, but I think at the pH of the cytosol, two phosphates like that are going to give you like three total charges, or 1.5 charge each based on the ionization of that phosphate. So you've got a whole lot of negative charges that are going to be sitting right here at the membrane, and that's going to change a lot of things. A lot of proteins are going to bind to that because they'll have positive amino acids and they'll come up and they'll bind to that and stick there. And between this mechanism and the next mechanism I'm going to talk about, it's going to involve turnover of PIP, PIP2. Okay, so producing it and breaking it down. Here in this first one, we're talking about producing it and how that changes the position of CAPZ. Here, this is CAPZ on the microfilaments. Plus end is blocked, so we can't polymerize. We need to open that up so we can polymerize. Now, I made a little animation of this years ago, and it's not the best animation in the world, but it's okay. I'd like to show it to you now. Bum, 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 bum. Let me just stop the share, find out where I put that. Okay. Pause. Act is. Share. Oh, chat's here. Somebody chatted me. Good morning, Brittany. Okay. All right. By the way, Brittany, if you want to ask a question or something, I'm not necessarily following that chat, so just yell out and interrupt me. That'll be fine. All right. Okay. So anyway, this is the animation. Here is your receptors, and here comes some hormones over here that are going to activate this. So hormone comes over, and this is part of what we'll talk about in 2 chapters from now, but that gets activated. PIK then makes PIP2, and those negative charges are going to attract the CAPZ, and CAPZ comes off. Now the plus ends are open, and we can start polymerizing. So that's part of what's going on here with this stimulation of the cell to start crawling in this direction. Activation of PIK, so we're making more PIP2, and that's pulling CAPZ off. Okay. So, now the other thing, and this is not an animation, this is just some static figures I made. The other thing that PIP2 turnover is doing is releasing some enzymes that are they're off right now. They're ARPs that are turned off because they're sequestered here on the membrane. And they've got nothing to do, so they're just hanging out here. So you've got your gelsolin cutting in the middle, profilin, and cofilin, ADF cofilin. These are all enzymes that are kind of turned off by being bound to PIP2. And then to release them, then we activate a different enzyme, phospholipase C. So phospholipase C is going to be really important 2 chapters from now. So I don't want to talk about it too much. But basically, back here in chapter 2, I talked about here's your phospholipid. Phospholipase C will cut this bond and release the phosphate and whatever the polar head group is here. So in this case, this polar head group is an inositol with 2 phosphates on it. So when you cleave this, you get an inositol with 3 phosphates coming off. And that'll be a signal transduction thing we'll talk about later. But what's most important here is you're losing PIP2. So when this comes along, it breaks down this PIP2, and that releases gelsolin, profilin, cofilin, and now they can go down and start the process. So you've taken the CAPZ off the top, and you've released these enzymes. We're ready to start changing our microfilaments around. And make sure I know where I'm going now. Well, I think I know what I'm doing. Does that instill confidence? So how does this happen? Oh, wait. What happened to my? Okay, so why are we doing this? Again, we're talking about changing the cytoskeleton, the microfilaments, in order for the cell to start crawling in a certain direction. So we're talking about probably our best example is something like a fibroblast that senses damage somewhere in tissue. And maybe, you've broken blood vessels, so platelets go in and they get activated, they release PDGF, and then the fibroblasts see that PDGF and they crawl towards it. PDGF would be activating these receptors. So what does the cell need in order to crawl in that direction? So there are 3 different structures it's going to need. There's another one that these slides throw in there just because it's something you should know. But we'll start out here with the Philopodia. So a Philopodium then, it sticks out from the membrane. It's really small. It's almost like a cilium, except it's structurally very different. But it just thing sticking out of the plasma membrane. Inside of it, you've got all parallel arrays of microfilaments. And that's important, the fact that they're parallel, because that's different from what we'll see in other places. So the plus end of the microfilament is embedded in some electron-dense material here, which basically just means proteins. So you've got the plus end embedded in the proteins and then parallel arrays. And these are going to be stiffened with cross-linking proteins. Oops. So here's your parallel arrays, and they're cross-linked with vilin and fimbrin. I think there's also in Philopodia, I know fimbrin's important in Philopodia. I think there's another protein called fascine. Let's not worry about that one. So anyway, you're cross-linking here. And by cross-linking them, you're going to make that a little stiffer. And then these are microvilli, which are structurally very similar to Philopodia. Microvilli, I've talked about them before. They're in the intestinal epithelial cells, increasing plasma membrane area. So you've got more transporters. You can absorb more nutrients from the gut. Anyway, these microvilli then are very similar to Philopodia, parallel arrays of microfilaments, cross-linked with vilin and fimbrin. And then we've got myosin 1 and calmodulin attaching it to the membrane. And that's kind of interesting. I mean, myosin, we often think of myosin as like a motor that's going to walk along a microfilament and produce motion. In this case, it's just being used as a structural protein because it binds to actin. All myosin binds to actin, but the tail of it then is going to bind to something else, and it becomes structural. And calmodulin, to me, this is totally ridiculous that calmodulin is here, because in my mind, calmodulin is always a single transduction molecule. It binds calcium and then it activates other enzymes. And we'll see that in a couple of chapters. But it is here as a structural protein. It's helping to bind the myosin and the microfilaments together. And then down at the base of the microvilli or the Philopodia, you've got spectrin, which we'll talk about later in this chapter, and some intermediary filaments, and that ties these up in this whole terminal web that holds them in place. And what these guys are doing is they're reaching out and they're sensing chemicals and they're making attachments for the cell. So they're the kind of the leading edge, the tip of the spear here of our lamellipodium. Another extremely important structure for crawling are the stress fibers. Stress fibers now are anti-parallel. So whereas, the other ones were parallel, these are anti-parallel. And that's really important because they will be cross-linked with myosin. Now here we're talking about myosin that will actually produce force and cause movement of these microfilaments. So you can see we've got 2 myosin dimers. They're connected by their tails. Each of them grabs a different microfilament and then walks towards the plus end of it. And that pulls those 2 myosin filaments together. That's going to be how the cell pulls itself forward. So it's going to put its lamellipodium out and then pull itself into that lamellipodium using these stress fibers. Another thing, now this is not necessarily part of cell crawling, but another thing that's really important are microfilament gels or actin gels. And, as we're looking at them here, they look like 2-dimensional structures, but they're really 3-dimensional structures. And all they are is just microfilaments, cross over each other. And then you've got filament, which is a dimer. It will bind to one microfilament and then bind to another filament, and then that binds to a different microfilament, and that's going to kind of like spot weld two crossing microfilaments together. And again, here, this kind of looks two-dimensional. But anyway, what this gel does, it's not necessarily involved in self-crawling, but what it does is it surrounds the whole cell underneath the plasma membrane. So this red here is your cortex. So the actin gel is what's used in the cortex, right under the plasma membrane to kind of support the plasma membrane and hold it in place. Here's cell cortex again, all the way around the outside of the cell. And all cells have this, which is why, your non-muscle actins are ubiquitous. You can use them as internal standards in protein gels because it's everywhere. This is just supporting the plasma membrane. Here's something I would recommend. This is Inner Life of the Cell, a fabulous animation. There's one point about a minute in where you go into the cell and you look back and you can see the spectrin network that's holding the plasma membrane together, which is something we're going to talk about shortly. And then you come into the cell and you can see this three-dimensional network of actin, and that's your cortex, your actin gel. So definitely look at that if you get a chance. Okay, so that's your actin gel, and then the third thing we need is a lamellipodium. So the lamellipodium is this branched microfilaments. Again, plus ends out at the leading end, minus ends in here. And the idea is we're like polymerizing out here and depolymerizing back here, forcing the monomers through so they can be polymerized out front again. So what's different about this is it is that branch structure. So we'll see, CDC42 coming along, activating WASP, ARP coming in here, and then branching it out. So that's our lamellipodium. Now, getting back to our G proteins, these are our Rho GTPases. You should know these. They're kind of important. So these Rho GTPases are what are going to be activated by the receptors in order to, these are part of the signal transduction that happens when the receptors get activated, and these go off and then stimulate ARPs to cause the changes in the microfilaments that we see. And this is kind of an elegant experiment. In your book, there's like, someone obviously took this exact picture and redrew it in blue. So it's like a cartoon of this. This is the actual experiment from this textbook that I've used before, World of the Cell. Here is a cell that's serum starved. So all the receptors are inactive. We turned off all the receptors. This is just kind of a quiet state of the cell. And then you can input a row that's constitutively active. So it's easy with these G proteins to change an amino acid, site-directed mutagenesis, change an amino acid so they don't break down GTP anymore. They're just constitutively active. So you take that constitutively active row and you stick it in a cell, and boom, the whole cell turns into stress fibers. You do that same thing with CDC42 and you get Philippodia everywhere. And if you do that with RAC, the cell becomes one big lamellipodium. So what this is telling us is when the cell starts to crawl, you're going to get activation of RAC, Rho, and CDC42. RAC is going to be responsible for producing that lamellipodium. Rho is responsible for making the stress fibers, and CDC42 is going to make the Philippodia. CDC42, as we've seen, is also involved in lamellipodium because it's going to cause the branching. That's a question I've always found a little bit confusing. I don't really know how they overlap, but just keep it in the back of your mind that that's the way it is. Sometimes in science, that's just the way it is. Okay, so again, what we're talking about here is chemotaxis. So chemotaxis is when you've got a cell that senses a source of a chemical and it goes towards that source. I'm walking my dog in the morning and we walk a little ways and he stops and he goes back and he sniffs here and he sniffs there, and eventually, boom, he finds the spot, finds that wonderful pea he wanted to smell. Chemotaxis, right? Sensing the chemical and going towards it. And again, the example I like to use is is fibroblasts moving into an area of damage. You've got, damage in tissue, and that damaged tissue is going to be releasing growth factors, especially from your platelets. The fibroblasts sense that, and they move towards the area to help correct that damage. So cell crawling. So that's what everything we've been talking about is leading up to, because you've got this source of a chemical. It's going to be activating receptors on the membrane here, and that's stimulating all these ARPs to start turning over the microfilaments and changing the microfilaments. And that's how it all happens. Now, what does it look like macroscopically? Is this macroscopic? Microscopic anyway. When you stimulate the cell to start moving in a direction, the first thing you see is the extension of this lamellipodium. Okay, extension of the lamellipodium, and we call that protrusion. So step one is protrusion. And this is how protrusion works. Basically, monomers are forced out, and that pushes the plasma membrane out, and then you polymerize them onto the microfilaments that are there. So it lengthens that microfilament, and that's pushing the membrane out. You're also branching here. It's too complicated to put it in this figure, but it's branching as well. Now, here's an interesting factoid. When this first starts to happen, we get what's called retrograde flow of actin, and that's because the microfilaments are treadmilling. They're going to be adding monomers at the plus end, and then the myosin is pulling the fiber back this way, and then at the minus end it's depolymerizing. And then you get the monomers being forced forward so you can keep doing it. So these microfilaments at first are not really growing. I think it's kind of like trying to figure out which way to go and getting ready to go, because it's sensed that it needs to move. Now it has to figure out where to move. So there's like this period at the beginning where you've got this retrograde flow of actin, which is treadmilling of the microfilaments. Got a really cool animation, not an animation, it's actually a video that shows this. Here it is. All right, not you, not you. Oops. Shoot. So Zoom puts this thing across the top of the screen, and I can't change tab until it goes away, and I have to sit here quietly until it goes away. It looks like it's not going to. All right, pull you down a bit. There, okay. Oh, shoot. All right, let me stop that over, start that over again. Things got weird. All right, let's get rid of this. All right. This is the one. All right. So when this cell, when it first starts to get stimulated, you'll see it's like ruffling at the edges. All this ruffling out here. Before it really starts to move forward, that's what's going on there. Music's a little weird, but that's okay. Start that over again. So out here, you can see the ruffling, and then this here is the stress fibers that are pulling the cell towards that direction. Okay. Now let me just pause this for a sec. Here's a cell crawling towards something. This is again your chemotaxis. So this cell senses a bacterium. Maybe it's releasing a toxin, and the bacteria is or the white blood cell sensitive to that. So you can see it going, finds the cell and eats it. Yeah, it doesn't last long. Let's pause for a second. Let's slow this one down. Play it at half speed. So you can see it crawling in a direction, of the bacterium, then it grabs it and it's over. It's over too soon, like life in general, all right? Okay, lamellipodium. So here's another little video of the lamellipodium. Let's do this. I think if I was to do it over again, I'd go into cell biology where I could use these fluorescent antibodies because it's so beautiful. Okay. No, I'm not struggling to take calculus because I quit math long ago. Okay, so here again is chemotaxis. He's trying to get that bacterium, or she is, they are, let's say. And eventually, oh, you can't avoid me. You can run, but you can't hide, bacterium. Got you. Got you. Okay. All right. So that's what we're talking about here. The main reason I showed you all that was because of oops, this idea here, which is retrograde flow of actin and this treadmilling. Okay, so that's going to go on for a little bit, and it's kind of like the car is revving up, but you haven't put it in gear yet. What's going to put this car in gear now is when you put down these focal adhesions. So focal adhesions are conglomeration of integrins, and these integrins are transmembrane proteins. They bind to it says substrate, basically fibronectin, and I think collagen as well. And then on the inside, they're attached to the microfilaments. So at this point, the myosin's pulling these microfilaments back and they're treadmilling, and then boom, it grabs onto the substrate. Now, when the myosin pulls it back, the filament doesn't move, the cell moves. So it pulls the cell forward into that. And then at this point, it just keeps moving by polymerizing at the front end and depolymerizing at the back end and pulling itself into that lamellipodium. So that is step 3. Step 2 then is attachment. Step 3 is translocation, coming forward. And step 4 is release at the tail. Now, anytime I've ever seen this figure, I've seen this exact figure basically in several different textbooks. They always show a little chunk of the cell being left behind. That doesn't seem very efficient for a cell. I think what that is when these cells are done in vitro, it probably doesn't work the same as it does in vivo, in that the connection between the integrin and the substrate ends up stronger than it would be otherwise. So, and that's why you end up with the chunk stuck over here. I don't think that happens in vivo. But anyway, bottom line, you got four steps here. You've got protrusion of the lamellipodium, attachment to the substrate, translocation forward, and release at the trailing end. So that's your cell crawling. And it's all based on, signal transduction we talked, and the repolymerization of microfilaments. What's going on here? I'm not able to change my screen for some reason. Oh, come on, baby. Oh, man. Is my computer hung up? I hope not. Okay. All right. So I'm going to talk a little about integrins here because they're really important for that cell crawling mechanism, but they come into play a lot later in the class as well. So it's best to just talk about them a bit here. So your integrins then are heterodimers of an alpha and beta subunit. On the outside of the cell, they've got this binding site for fibronectin. Again, I think also collagen, but I could be wrong about that. But basically, extracellular matrix proteins they're binding to. And then they pass through the membrane. And now on the inside, they've got a binding site for intracellular proteins that are going to connect it to the cytoskeleton. You can see it here. This is from your book. I like this other figure better. Here's your extracellular matrix proteins. You're bound to them here. This then is talin and vinculin. So these are really important for attaching those integrins in a focal adhesion to the cytoskeleton. And then this is alpha actinin, okay, another important protein here, and that's cross-linking the microfilaments. And so myosin would be pulling on these microfilaments, and they're attached to the extracellular matrix there. All right, so this is what the focal adhesion looks like. And basically, it's like a spot weld. It's like a little foot sticks down to one place. So you don't have integrins everywhere pulling it along, but they're all focaled into an adhesion, thus the name. All right, so this next slide, this isn't that important here, but we're going to talk about it later, and it's really important later. So I'm throwing it out here as long as I'm talking about focal adhesions. These, in addition to being structural, are also signal transduction complexes. They're really important for signal transduction. And that's so that the cell knows it's attached to extracellular matrix. Why is that? Well, normally if a cell isn't attached to extracellular matrix, it'll go into apoptosis, right? That's a protection against metastasis. When a cell releases from a tumor, typically it's going to go into apoptosis unless it's found a way to get around this mechanism. So the basic idea here is when the integrins are attached to ECM, we've got kinases here, SRC and FAK, focal adhesion kinase, that get activated. That turns on signal transduction, which is going to inhibit your apoptosis proteins and allow the cell to stay alive. SRC, by the way, interestingly, is the very first oncogene that was ever isolated. It was isolated from the Rous sarcoma virus. So you can imagine a retrovirus going in, putting itself into the genome, accidentally grabbing a kinase and coming back out, and now it's an oncovirus instead of just an annoying virus. So that's kind of the evolution of the Rous sarcoma virus, which is, you're totally safe. It's only in chicken. It's only infects chickens, not humans. So Rous sarcoma virus, that's where the enzyme or the kinase SRC got its name. SRC is also extremely cool because it's we'll get into this later, but why not talk about it now? When you look at its structure, it's got 3 domains, and they're called SRC homology 1, SRC homology 2, SRC homology 3. And SRC homology 1 is just the kinase, tyrosine kinase. And then you've got SH2 and SH3 that are important in sticking proteins together. They're like adapter domains that stick proteins together. And they're similar in a lot of different proteins. So we see SH2 and SH3 domains in a lot of different places. They were first discovered here in this enzyme SRC. SRC. And I just keep talking about because I like saying SRC. Okay, so that was cell crawling. We're all done with cell crawling. Don't confuse that with this. This is the other type of non-muscle cell movement, which is a lot simpler, I think, than cell crawling. And it's just amoeboid movement. Amoeboid movement involves depolymerization of microfilaments in the back, here it's turning from fibrous actin, F-actin, into globular actin, G-actin. So these G-actin monomers then are forced through the center of the pseudopodium, and when they get out here, they push the membrane out, and then they polymerize and solidify, basically, the membrane once it goes out. So that's the way your pseudopodia are. Oops, I seem to be missing a slide here. Okay, that's the way your pseudopodia are formed. So it's a much more simple type of movement than cell crawling. Let's watch some of this. I think this would, okay, let's not do this. We just, well, maybe. I think this might be, is this amoeboid or is this cell crawling? think this is amoeboid because it looks like the cytoplasm's kind of streaming. You don't see one big lamellipodium, but just kind of pseudopodia sticking out in different directions, looking for somewhere to go. This music is thrilling. Must be exciting to be a neutrophil. It's not the best. And let's just do amoeboid. Amoeboid movement. Okay, so here's a 30-second amoeba. Here's your 30-second amoeba. So there's you can see cytoplasm and stuff in the cytoplasm flowing into that pseudopodium. And in addition to that, whatever those big chunks are, they must be organelles. There are G-actin going out there and then polymerizing. Because it flows out and then it gets hard. Okay. Okay, people, let's stop this share. All right, so I guess that's about all I wanted to do on yesterday. On Wednesday, I started talking a little bit about myosin, but it doesn't make sense to start into that. Let's just save that. And then save the myosin. We'll do all of that myosin in the next class on Monday. We'll do myosin, skeletal muscle contraction, and then skeletal muscle EC coupling, cardiac EC coupling, smooth muscle EC coupling, which is crazy. And then hopefully we can finish this chapter by Thursday because our exam's the following week. If I don't get through all this material by Thursday, then we'll just put the exam off for another few days or something. Right now I've got the exam scheduled for a window of 3 days. Please let me know if that's okay. Someone suggested that because since you're not synchronous, you shouldn't necessarily have to be taking the exam all at the same time. But let me know if that 3-day window works for you guys. I can always change that beforehand. Anything else? Anything else? I think that's all I got. I'll post this video probably within the hour, and then you can watch it over and over and over. It's an excellent lecture. You don't want to miss this. All right, so that's all I got. I want to say goodbye, Brittany. Bye, Brittany. And bye, everyone else who's watching it as a recording.
Bye, Dr. Slish.
Love you all. Peace out.
Bye.